Well, thank you very much for the opportunity to speak to you today. So when I first was given the invitation to repeat what was a 25-minute talk in Cambridge to commemorate the life of Peter Armitage, who had died last year, I wasn't quite sure how best to sort of fill the time. So I thought I'd place the work in the context of things I'd done before and since. And now my concern is more whether it fit nicely within a 50-minute time slot. Just to give you an outline. So I'll give first of all a bit of background. So how I came to start working on this topic, which my first post as an epidemiologist was working in the cancer epidemiology department, and I'll say a bit about that. I'll then describe multistage models, what they are, the mathematics behind them, and explain some of the biological insights that one can gain from modeling data in that way. I'll then move on to multimorbidity, explain what the challenges are there, and mention some new developments about how you might identify when there is a shared step that's necessary for progression or development of more than one disease type. And then I'll give some of my personal reflections on the future of epidemiology and public health. And that last part will take 4 or 5 minutes or so. So background. So I was originally a theoretical physicist, and I did a MSc in applied statistics in 2015, and then joined the cancer epidemiology unit at the University of Oxford. And whilst I was there doing my analyses using typically proportional hazards methods, I started thinking, how could we improve the methodology by thinking about the processes that were occurring in terms of the development of cancer. Try to admit somebody. And the first thing that occurred to me is that if cancer is occurring due to mutations in cells and cells going wrong, then surely cancer risk could be proportional to the number of cells at risk. It turns out lots of people have gone through this exact same thought process, and there's something called the Peto paradox, which is to do with this. And around about 2015, just when I was thinking about all these things, this paper appeared, which showed that you could broadly explain the variation in cancer risk in different tissues within our bodies just by consideration of the number of cells and the rate of stem cell division within them. So that was really interesting and got me started following a whole sort of train of research articles that I'll mention later. The other thing that occurred to me is, well, if there was a sort of a biologically inspired model, then surely we could both model the data better and gain some insights from doing so. And so that led me quite quickly to the multistage theory of carcinogenesis, which has been around since the 1950s, and I'll be saying more about shortly. And then the final thing that was going through my head whilst I was sitting there working through these new big data sets that are arriving, that we've got these new big data sets and we've got these new machine learning methods. And there were these possibilities that were never there before. And so in particular, I proposed this fellowship project, which was to address the question, given these new big data sets and these new modern statistical methods, what would a data-driven classification disease look like? So once you start thinking about how you define a disease, it's not as straightforward as you might think. So you typically have a collection of symptoms, and you need a sufficiently distinct collection of symptoms to distinguish one disease from another, but you also need a sufficient number of cases for a meaningful statistical analysis. Now, interestingly, if you go right back to the 1600s, to what I regard as the first sort of epidemiological study, or certainly the first sort of big data study, by a guy called John Graunt, who was around at the same time as Isaac Newton, you could see very similar sort of issues arising there. So what he did is he took the bills of mortality that were collected in London. The purpose of collecting this data was to spot them when there was an outbreak of plague, but it also gave information on the causes of death. And what John Graunt did was sort of tabulate this information across many different years so that he could understand what was actually the genuine sort of most serious causes of death, as opposed to those that were just sort of catching people's attention at the time. When addressing this problem, the first thing I thought of was, well, what information do we have? So we've got a lot of information on risk factors. So there's conventional information such as, BMI, smoking status, etc. There's genetic information, there's information from blood tests, and there's also these hospital diagnoses of diseases, which gives you information about the age at which they're diagnosed. The co-diagnosis, that's so you go into hospital for one condition, they make a series of tests and they find you've got a whole string of other things that you didn't realize previously. And you get these sequences of diagnosis that can occur over the lifetime of a person. And I realized that lots of people were looking at these sequences of diagnoses, and they still are actually. The genetic analyses takes about typically 6 to 12 months before you can really get familiar with doing those sorts of studies. So I postponed that. And the first thing I focused on was age at diagnosis. And I wanted to cluster diseases, sort of formulate a kind of definitions of diseases by clustering, but it needed to be meaningful. So I wanted to be able to understand why a particular disease was assigned to a particular cluster, and I wanted to understand the characteristics that are shared by diseases that were there. I was also keen to sort of build upon existing epidemiological methods. I didn't want to just sort of start from scratch. And so what I did was I started clustering using age of incidence, and I used a simple parametric multi-stage model of disease because this provided a sort of an understandable method. It was when you've got a parametric model, you can extrapolate forward in age, and it provided some biological insight. So what is a multi-stage model of disease? So here's the basic idea. So going back to the 1950s, the model that was conceived was of cancer originating through a sequence of genetic mutations. So you'd remain healthy until that very last genetic mutation occurred, and then the cancer would develop. And what the authors realized was that if these mutations occurred at an approximately constant rate, then this would lead to a very particular distribution in the incidence of cases as a function of age. And so they realized this, and they realized if they plot the data on a log-log scale, you should see a straight line, and that the slope of that line would give you information about the number of steps that are needed to trigger the cancer. And this seemed to work pretty well, and it must have been very exciting at the time. But since then, the model hasn't really got that much use. And there's some possible reasons for this. And one is it would have been, certainly in that period in time, difficult to formulate and to understand. It would have been difficult to fit the data very often. There are serious issues about whether it's actually identifiable, whether you can be certain that that is the process that's the model is fitting. But I think more importantly, alternative methods came along, such as logistic regression and Cox proportional hazards, which were just more suitable for a lot of the questions that people were interested in. But as George Box famously once said, all models are wrong, but some are useful. And this is a model that history has shown has proven to be very useful when it has occurred. So when it has appeared in the literature, so in the 1970s, the early 2000s, it's been associated with substantial breakthroughs in our understanding of cancer processes in this case. So retinoblastoma, colorectal cancer, and myeloid leukemia. But it was really sort of in the mid-2010s when there was an explosion of interest on this topic of multi-stage cancer processes. And I think this occurred because of the ease with which one could start to sequence the genetic information in these cancers and actually experimentally determine what was going on. And I do recommend you taking a look at these these articles. They're all beautifully written and absolutely fascinating. And amongst these publications was what I regard as quite an important discovery, which and this was reported around about 2019. So what the authors did was they looked at the blood in older people, and what they found was it's very common to see a portion of that blood which is made up of a clonal population, so of what are effectively cells with mutations in them that are distinct from your normal healthy blood that you've lived through with most of your life with. And these clonal populations were seemingly harmless. They're potentially precancerous, but they're nonetheless there and increasingly common as we age. And subsequently, in almost every tissue that people have looked at, or as far as I know, every tissue that people have looked at, so in the throat or in the skin, the similar things have been found. Now, with hindsight, this perhaps is not any great surprise, because if we look at our skin, I mean, most of us have got moles. These become more common as we age, and if they start changing, well, you've got to go to your doctor to assess whether it's a change that's potentially cancer causing, or whether it's harmless. And if it is a malignant transformation, then they can just give you a local anesthetic, remove it, and problem's gone away. So this sort of raises the question: if we're as we age, we're increasingly our body is a growing number of these sort of mutant clones throughout us. Are these related to other diseases? Are there other sort of multi-stage diseases out there? And there's been certainly quite a bit of work on neurological diseases, amyotrophic lateral sclerosis in particular, or ALS, where there is sort of a growing evidence that the disease is consistent with a multi-stage process. Most of these studies, however, involve an analysis a bit like this. So you plot your data on an appropriate log-log scale. You find that there's a straight line, and you say, well, you infer that it's consistent with a multi-stage disease process, but you can't really say too much more than that. So what is a multi-stage model in its most sort of general sense? So a multi-stage model is any model where you go from a healthy to a disease state by one or more independent pathways. They have to be independent, and through either a sequence or steps, or and they can be either required to be sequential or non-sequential. Doesn't matter. When that's the case, then you can write down a mathematical equation or the probability density function. However, that's all very nice, but in practice, for most diseases in most people, if there would be a pathway. Sorry, let me start again. In practice, there will typically be one pathway by which the disease occurs more rapidly than the others, and that will dominate your observational data. And when you have this situation, then for most diseases in most people, you can approximate this with what is in practice a Weibull distribution. So a Weibull distribution has a proportional hazards part with a relative risk, and it's also got this instance whose cumulative hazard function goes as h to a power. So how common might these multi-stage diseases be? Now, bear in mind that just for that model to hold, you just need a sequence of rate-limiting steps. It doesn't have to be complex genetic mutations, although that is one reason why you might want to look at this question. So I looked at 400 diseases in men, 400 diseases in women in UK Biobank, and these are just sort of randomly selected examples, just to sort of emphasize that there are lots and lots of these things that fit the data very well. And about 60% of the diseases that I looked at were consistent with the model. It would probably be less than that if you had more data, because that would put stronger constraints on the model. But nonetheless, it's a large proportion of the data, which sort of raises the question: well, why hasn't anybody noticed this before? And I think there's two reasons for this. I mean, one is simply that the data wasn't available. in such large quantities. And the other is you have to do the left truncation correctly, and this is a bit tricky. So typically, what you have in a cohort study like UK Biobank, you don't collect the data over an entire lifetime. You recruit patients from an age range of typically around 40 to 60 years old. Now, by that point, some people will already have experienced the diseases that you're interested in studying. So the survival function will be less than 1 already. So if you were to plot your data on this log scale to look for see whether this was consistent with a multi-stage disease process, if you have data over the entire lifetime, then you see this lovely straight line. So this is simulated data. But if you only have data from age 40, you'd see something very different, which looks nothing like a straight line, and you would quite reasonably, but wrongly conclude that was not consistent with the multi-stage disease process. However, if you make an adjustment to correct for the fact that you've left truncated in this way, and I show how to do this in the paper, then you regain this lovely straight line, and you form a very different conclusion to what you would originally have had. To make this correction, you really need a parametric model. I don't think you could do this with a Cox model. It's not very easily. Okay. So going back to the George Box quote, all models are wrong, but some are useful. So the most useful thing about this model are the insights that it provides. So once you take several hundred diseases and you start trying to cluster them by their characteristics, several things start to stand out. One is that whereas some diseases are sort of a low risk throughout your life, and this risk increases slowly with age, there are these other diseases for which the risk is negligible for most of your lifetime, but then it really increases rapidly towards the end. And those sort of late onset diseases are typically ones which are more serious. On the other hand, the more sporadic diseases are the ones which are placing the strongest burden on the National Health Service. So they're the ones that you might want to avoid to keep people healthy. Another concept that drops out of this way of modelling disease is an effective age. So if you take this Weibel model and you take the relative risk and you place it inside the brackets there, then what you get is an effective age. So if your relative risk is bigger than 1, then your effective age is greater than your actual age. On the other hand, if your relative risk is less than 1, then your effective age is reduced. And this is useful because if you were looking to screen people who are in at an equivalent risk of disease, you could use that effective age to minimize the risk of over or under diagnosis, because you don't want to diagnose people with a disease which and leading to unnecessary procedures. But on the other hand, you don't want to miss anything that's potentially serious. So it's important to get that to optimize screening. You can also interpret this factor as an aging rate. So you'll notice that by putting the relative risk inside the brackets, this power of 1 over k has come in, and that k is related to the it's the power that we saw earlier. So the sporadic diseases tend to have lower values of this k, and meaning that in terms of a relative aging rate, they're much more modifiable. So not only are these the diseases that are placing the highest sort of burden on the NHS, they're also the sort of diseases that are the most modifiable in terms of risk. On the other hand, these late onset diseases, which have a higher value of k, because that suppresses this the effect of the relative risk, they're much less modifiable, and the risk continues to increase quite rapidly with age. Another thing you can do is you can use a strong risk factor, such as a genetic risk factor or something such as smoking, as I'll show in this example, to stratify your data And then by comparing the differences in the data, you can make inferences about what might be going on biologically. So to look at this example here, we've got the smokers in red, the non-smokers in blue. On the left, there's this sort of rigid displacement of the curve upwards, and that's just consistent with smoking increasing the rate of the processes that are leading to disease. So it's the same sort of thing as you'd see in a proportional hazards model. In the middle here, the red curve has moved upwards, but it's also reduced in slope. And that's exactly what you'd expect if smoking was increasing the rate of the processes in one of the steps to such an extent that it's no longer rate limiting. So you're reducing the number of steps in the disease process. On the far right was something very different, which was difficult to explain initially, but I have a proposal as to what's going on there. So there, what is peculiar is that the rate has increased, but so also was the slope. I don't know, has anybody got any ideas what's going on there? So it turns out that the type of cancer experienced by smokers is different to that predominantly seen in non-smokers. So what I think is happening is that smoking is increasing the rate of this particular cancer type sufficiently that whereas before it was making up a small part of the observational data, now it's being swamped by it. So I think what we're seeing there is effectively a different disease. But the reason why I looked for that was because I was trying to explain what was going on in that data set. Okay, so that brings us on to multimorbidity. So there's a recent paper on multimorbidity which was claiming that it could be explained by multistage disease processes of the type I've just described. Now, I think it's likely that some of these processes will be able to be explained by multistage disease process, but probably not all of them. But nonetheless, I'm just bringing it to your attention in case you're interested in this. So what is multimorbidity? So multimorbidity is a word used to describe multiple coexisting diseases within an individual. It's very common and it's becoming more so. And we don't know the best way to treat people with multiple conditions, and we don't even know how to study them. And it requires a much greater sort of general understanding of diseases and the interactions with medications and treatments. So, so I was thinking about this from the perspective of clustering diseases by how modifiable they were, and at this point now starting to look at how other diseases might change your subsequent risk of a different disease. And thinking about how to do this led me to raise the question, well, how much disease risk is simply due to old age and established risk factors? So here's the problem. So epidemiology traditionally studies diseases independently, one at a time, in people whom are otherwise pretty healthy. So this is the setup you typically have. So you're born. At some point, you're recruited into the study, so UK Biobank, for example, and you're followed until you either experience the disease that you're interested in studying, or you die or have a serious disease such as cancer, at which point you would censor the individual. However, what you have in practice is something more like this. You're born. You have several diseases, you may have accidents, you may have something serious, who knows. You then are recruited to the study, during which period you have many different diseases, various different treatments, all of which are potentially interacting with each other. So the question is, how do you study this real situation? Okay. Well, what occurred to me was that there are some things that you can do in a fairly sort of rigorous way from an epidemiological perspective. So you can estimate adjusted disease risks in men and women whom are otherwise disease-free, so otherwise healthy. You can predict then disease risks in people who would be otherwise healthy, otherwise disease-free. You can estimate the expected number of new disease cases within that cohort that you're looking at, and you can compare it with what you actually observe. And then you can get some sort of indication of how much the disease risk has increased above that you'd expect in otherwise healthy people. And the only thing there that is not in a textbook somewhere is this bit, which is how do you turn these risks into expected numbers of cases. And for that, I'll introduce the Poisson binomial model. I don't know if anybody here heard of it at all. It's sort of the only model, you know, if you see these figures where they show all the different probability distributions and how they all relate to each other, I've never seen this one on one of those yet. It's quite recently been sort of explored, but it basically involves a product of sort of Bernoulli distributions and an indicator function that ensures that the total number is equal to the total number of diseases that occur. So these probabilities, you would just estimate these with conventional survival analysis methods. So it would be the probability of a disease in the study period in someone that's otherwise healthy and with adjustments for whatever risk factors they may have. The properties of this model are comparatively easy to work out. The main properties are comparatively easy to work out. These are the two things you really need, the mean and the variance. There's a generating function that you can work out fairly easily, or comparatively easily. And if you take the limits of these probabilities becoming small, what you find is that this tends to this generating function, which is the generating function for a Poisson distribution. So that's presumably why it's referred to as the Poisson binomial model, because in the limit of small probabilities, you get this Poisson distribution. Okay, so then it's just a matter of doing a conventional epidemiological study. I won't go through the details, so it's just the standard sort of thing. You define your diseases carefully, you exclude people who've had serious diseases before the start of the study, you left truncate at the time they start, and you censor if they get a serious disease, and you adjust for all the well-known risk factors, smoking, BMI, socioeconomic status, etc. And you get your estimates for people who are otherwise healthy, really. Okay, and then what we do is we compare the predicted number of disease cases in this cohort, which is the UK Biobank cohort, with what is actually observed. So the predicted cases are in blue. So we've got the predicted cases in blue, the observed in black, men on the left, women on the right. The blue points line, this nice straight line as they should do. That's just the test the model is working. Now, what I expected to see was quite a sort of smooth distribution of points above this blue line, quite a variation. But actually, the increase is about 50% or so for most of these diseases. Now, if you break it down by sort of classification of disease. So according to the International Classification of Diseases version 10, the diseases are sort of grouped into various chapters, as they refer to them. So you've got disease of the circulatory system, disease of the digestive system, musculoskeletal system, etc., etc. And when you break it down in that way, there are differences. But the increase is still typically somewhere between 1 and 2. So it's actually, well, first of all, it's not that big. And it's also fairly similar. So if these results were to generalize, then what you can infer is if you just reduce the underlying risk of disease through changes in lifestyle, so you follow the advice from conventional epidemiological studies, that will pull down the number of diseases you'll observe. And we might also expect to pull down the overall total number of cases occurring in even in the more complex situation where people are not healthy at the start. So if these results generalize, then first of all, we get this important result that reducing the risk of primary disease reduces the overall disease risk. So that's basically saying these conventional epidemiological studies are worth doing. There's scope to build a phenomenological model to predict demands on healthcare, because you can just sort of get an estimate for how much the actual number of cases are expected to be above what you predict. And disease risk will often be modified more by these sort of recognized risk factors that we already know and understand than by these very complicated disease or multifactorial interactions involving medication and treatments, which is encouraging. Now, there are some limitations here. So the study considered the first disease in each ICD-10 chapter. So with the first disease in, say, cardiovascular diseases, or the first disease in cancers, etc., not the first disease in an individual. So that's just quite a weakness. I needed to do that to ensure there was enough cases in the study for a meaningful analysis. I only considered diseases that were common enough and could be modelled sufficiently well with this parametric Weibull model. And the big question is will these results generalize to other cohorts? So this really needs to be addressed, but it has potential, quite a lot of value in terms of the applications going forward. So this brings us on to a simple model for multimorbidity. Because as I said, I was quite struck by the way this increase is fairly similar across many different diseases. I say this is about 50%. So if you want a rule of thumb, that's not a bad one. And I started thinking, well, how could you explain this? And one possible explanation is if there are a smaller number of shared pathways that are influencing many different disease types. Okay, so I played around with the maths of this for quite a while. And it was only at the Armitage workshop last year that it suddenly dawned on me there's something very important that comes out of this way of thinking that actually is pretty simple. It doesn't really require much mathematics at all. So here's the model. So let's assume that there is some sort of shared step leading to two or more disease types. So this step itself may involve many separate individual steps and many different complex processes, and the subsequent diseases may require several other steps before they occur. But let's assume that they share a step, okay? Now, in contrast, the diseases could be independent. So let's imagine the situation also where you've got exactly the same thing, but the two diseases now are independent. Now, if there's no prior disease, if you're looking at the first disease you observe in an individual, you cannot tell the difference between these two models. The incidence rates will be identical for, say, disease J and disease K in both these models. On the other hand, the moment that you observe one disease, say disease J, then if the situation is this model, then the rate at which you will observe disease K in subsequent observations will be much higher, and you can use that to indicate the presence of a multi-stage disease process. So, rather there being a shared step. So just to illustrate this, so here is the both models, the instance data for the first ever disease. This is simulated data, and the lines are on top of each other, so you can only see the red line, which corresponds to if the diseases were independent of each other. And then this is the data if you are looking at the cases after you observed one disease. Now, if the diseases are independent, you get the red line, which you can see is the same slope. Now, to do this analysis, again, you need to do this this corrections for the left truncation, because you're now looking at cases beyond a certain age. But that's not a problem. On the other hand, if you've got this model with a shared step, then you see something very different, and it's qualitatively different. Not only has the rate gone up, but the slope is reduced. If you didn't, without that reduction in the slope, if this rate had gone up, the slope wasn't reduced, you would say, well, disease J is modifying the risk of disease K, but it's not sharing a step. On the other hand, that reduction in slope is strongly suggestive that there could be a shared disease process that's occurring. So this is a comparatively simple statistical analysis that one can do to provide evidence for two or more diseases sharing a common process. So that brings me on briefly to what I'm doing now, which is working on the Compute project, which is funded by NIHR, which takes another approach to studying multimorbidity. So I think the underlying idea, which I think several groups and people are starting to think along these lines now, is that a situation like this where you've got sort of aging and exposures on the one hand, acting through some sort of shared mechanisms or pathways, leading to various sort of clusters or patterns of disease. And the reason why this picture is appealing is not just because it's plausible, it's also because if this is the case, then you might be able to find ways of sort of targeting these pathways with a drug or a treatment to sort of reduce a whole group of disease types, a bit like with a statin, for example. So I'm working on the trajectories model, which is led by David Steinsaltz in the statistics department. The whole project's led by Rafael Pereira and Claire Bankhead. And it's sort of, we're a way off. Well, we're going to start producing results within the next few months. The models are sort of just at the point now where we're starting to apply them. And the basic idea is you have these sort of sequence of measurements in an individual that are collected by GPs. So, for example, systolic and diastolic blood pressure, which are the crosses. And some of those changes will be sort of noise. Some of those changes will be temporary, and then it'll recover. But other aspects will reflect underlying physiological change and changes in disease risk. And the idea is that with a suitable model and these sort of sequential Monte Carlo methods that we're using to fit the data, that hopefully we can identify these sort of latent trajectories that are provide a better explanation or a better measure of disease risk than the raw data itself. So that's work that's ongoing, and hopefully if all goes well, I'll tell you all about it at some point in future. Okay, so I'm just going to finish up with a few of my own reflections on epidemiology and public health research. So I don't know if there's any epidemiologists in the audience, but I regard epidemiology as the science of doing statistics and getting the right answer. So if you've ever dealt with medical data, if as a new statistician you were to start analysing medical data without the background of epidemiology, you would very likely go very wrong. So another way I like to describe this is, it's how to do better than a graduate statistician without knowing very much maths. And I believe this is true. And let me say a bit more about this. So why does the epidemiologist tend to do better with medical data? Well, they gather all the available understanding about the disease, and they build it into the model. So for example, you would exclude people who've had something seriously wrong beforehand. You would censor if somebody gets a disease, a serious disease that will modify your risk. And then they go on and they estimate the strength of association between the risk factors and the disease. Now, I was originally a physicist, and when I think through this process, it strikes me it's very much like theoretical physics. So what does a theoretical physicist do? Well, you gather all the available understanding about the causes of a phenomenon, you build it into your model, and then you explore the consequences of that model. And this is something which, to some extent, is not yet being done by epidemiologists. And I'll say a bit more about that. So causal assumptions are something that are necessary to consider for a sensible statistical analysis. So this is the causal diagram is effectively the epidemiologist's model. Now, to give you an example, so let's imagine you're interested in the influence of BMI on the risk of some disease. You've got some set of confounders. They might be smoking, education, socioeconomics, all the usual stuff. And your boss comes to you and says, Anthony, the disease you're interested in its risk is modified by blood pressure. So I think you should include blood pressure in your model. Sorry. Okay. Who thinks I should include blood pressure in the model? Any yeses? Any no's? Any more nuanced answers?
Yeah.
Well, I mean, it depends. If you're interested in, so let's see, if you're interested in the effect of BMI on the disease, and not, so blood pressure will no doubt in this model, it would mediate some of the effects of the BMI on disease risk. So if you're only interested in the influence of BMI on disease risk, you probably don't need to have it in the model. On the other hand, if you're taking the view, well, blood pressure, we've got drugs for that. I'm more interested in the underlying cause influence of BMI on disease that's not mediated by blood pressure. Well, then you would want to have it in your model. So it
also rather depends on the timing of these things.
Okay.
So
Yeah. So what I'm saying is that you need to make a decision at the outset as to what you think is happening to set up a sensible analysis. And this holds even if you're trying to interpret an analysis. So there's a paper that's been rewritten on this that I'll refer you to. It's been around for well, at least since 2013. It's called the Table 2 fallacy, and I do recommend taking a look at it. And it points out that very often in a lot of these studies, on the Table 2 contains a list of all these associations, but the interpretation of them should be very different, and it depends exactly on how you're thinking of the model in practice. So what I think is going to happen over the course of the next decade or so, these changes often happen very quickly when they do occur, is I can imagine epidemiology becoming a lot more like sort of theoretical physics in terms of thinking in terms of models. So I can imagine you're building your model and you'll clearly state it in the form of some sort of causal diagram. We'll then deduce the consequences through statistical analysis or mathematical study, and then you update the model. So you learn a bit more about the disease process, you realize that model was wrong, you change it. Okay, and that's what you do in physics all the time. You build a model and you test it to destruction and you update it. And the great thing about that process is that once you've assumed the model, there are other quantities you can estimate, right? So once you've got this causal diagram, you can start using the methods from causal inference that have been developed over the last sort of 15 years or so, 15, 20 years. You can ask questions such as, if everyone in the population stopped smoking, how much would disease incidence change? And you can calculate things such as population attributable fractions, average causal effect, etc. You could ask the question, if you stop smoking, how much will your disease risk change? And this is characterized by things such as effect of treatment on the treated. Or another question that you see in legal situations is, let's say, given that you've died from lung cancer, how likely is it that this would have occurred if you had not smoked? And this is referred to as the probability of necessity, or the probability the event would not occur but for the defendant's actions. And once you have a causal diagram, you can calculate these quantities. Whereas at the moment, all we calculate are relative risks, which only convey a much smaller amount of information. So what about public health? So last two slides. So Richard Dole famously once said that death in old age is inevitable, but death before old age is not. Okay. Well, I think I agree with him about the death in old age is inevitable. I don't see much way past that. But I think as an ambition for the next sort of 50 years or so, perhaps ill health is not. And if we can sort of identify the main causes of poor health and eliminate them, perhaps that's what we should be aiming to do over the course of the next 25 to 50 years. So at that point, I'm going to stop. I'm going to say thank you very much for your attention. Sorry if I've gone too fast. Thank you to the funders for allowing me to do this work. It's been an absolutely fascinating 10 years, and I've thoroughly enjoyed it. And if you have any questions, please do send me an email. I'm always happy to talk about science or look up some of the references. So thank you.
Thank you very much. Wonderful. Do you have any questions? I was interested in one of the areas that you didn't mention in detail. It seems to go right back to the beginning, and that was whether or not there was consistency in the number of steps required to explain particular diseases, whether that varied. Clearly, it varied with the curves, but I wonder if that's a feature.
So I think I understand your question. So what you're asking me, if I understand correctly, is whether when I look across the range of all those different, several hundred diseases, whether there are groups of diseases which share 5 steps, or groups share say 7 steps. And that was exactly what I wanted to look at. And I was hoping to see, for example, spikes perhaps that indicate the integer values, perhaps if there are a certain number of rate limiting steps. It is possible that is the case, but more data would be required because the confidence intervals are too wide to really be to see certainly differences of an integer or so for most of those diseases. So what I found was more of a continuous spread. So whereas I was hoping to cluster diseases using these parameters, in the end I thought clustering was inappropriate because it's a continuous spread. I just split them to tertiles. So then I had my sporadic diseases, which are common early in life, our late onset diseases you don't see until very late in life, and then stuff in the middle, broadly speaking.
Yeah, it's a fascinating synthesis in many ways. One wonders to what extent the clustering owes a lot to the common risk factors in its structure, and how you could investigate that.
Well, so that was the other question. So how much of this clustering of diseases might be due to shared exposures? So this group of individuals has experienced a certain, the smokers, for example, may have an increase in a certain set of disease types, etc. So the first paper I actually published on the clustering clustered diseases by association between the risk factors. So it was quantitatively, it was using the associations with risk factors and their covariances and using those properties to cluster the different disease types. And yes, you can get some very sensible looking clusters that appear from that approach. What I had been hoping to do was to pull together all of my studies into a final piece, which produced this is my classification of diseases using all these various subparts. But I never got to do that because I ran out of time, partly due to a serious bicycle accident, partly due to COVID. So just bad luck, really.
I'm particularly interested in beyond cancer. There was a graph there that you had around Parkinson's, and I also saw certain nationalities on there. Where do you see, I suppose, this research being able to help and focus in on active, if you like, engagement at certain times before the diseases, the final diseases start presenting themselves? So the interventions.
Yeah.
So is there are you seeing applications of that already, or
well,
hoping to see?
I mean, there are hypotheses that some of these neurological diseases, for example, might share common processes.
Sure.
Well, we can test that now, I think. I think you need enough cases, but that might be testable with the approach I just outlined. There are lots of other diseases, again, for which you might want to determine whether there are any sort of shared steps. Because so going back to your question, so for neurological disease, if you determine there is a shared underlying process that's responsible for, two or more of those diseases, well, then you would really focus your effort on determining how to treat that. In much the same way that you can take a statin to sort of reduce the risk of several disease types, which of course would be in everybody's interests, because it would be in the public's interest to reduce your risk of disease, and in the drug company's interest, this drug that they could sell to half the population.